Glycolysis is the first stage in the breakdown of glucose for energy production and happens in the cytoplasm of cells. It is a major mechanism in cellular respiration present in both prokaryotic and eukaryotic organisms. One glucose molecule is transformed via glycolysis into two pyruvate molecules, producing a negligible amount of ATP and lowering power in the process. The glycolysis process consists of 10 enzymatic processes, each catalysed by a different enzyme. The first step is the phosphorylation of glucose, which is accomplished by using an ATP molecule to add a phosphate group to glucose, resulting in glucose-6-phosphate. Depending on the tissue, either the enzyme hexokinase or glucokinase catalyzes this process. What is hexokinase and glucokinase? Hexokinase is a component of general glucose metabolism and is present in the majority of tissues. In mammals, there are 4 hexokinase isoforms with hexokinase 4 also known as glucokinase. Hexokinase can phosphorylate glucose even at low glucose concentrations because it has a high affinity for glucose. It is frequently found in tissues like the brain, muscle, and adipose tissue that use glucose extensively. Under typical physiological conditions, hexokinase functions at close to maximum capacity and is not significantly influenced by hormonal or metabolic variables. Glucokinase is mostly located in the liver and beta cells of the pancreas. In contrast to hexokinase, it functions at higher glucose concentrations and has a lesser affinity for glucose. Because of this unusual characteristic, glucokinase can function in these specialized tissues as a glucose sensor. The liver cells can sense blood glucose levels thanks to the enzyme glucokinase and change their metabolic processes accordingly. Glucokinase is an essential component of insulin release that is triggered by glucose in pancreatic beta cells. In pancreatic beta cell, glucokinase functions as a glucose sensor in pancreatic beta cells, helping to control insulin release in response to variations in blood glucose levels. Glucokinase in pancreatic beta cells metabolizes glucose when blood glucose levels rise, increasing ATP production. The closing of ATP-sensitive potassium channels, membrane depolarization, and subsequent opening of voltage-gated calcium channels are some of the actions brought on by this increase in ATP levels. Calcium overflow causes the beta cells to release insulin into the circulation. In the liver, the principal glucose phosphorylating enzyme in the liver is glucokinase, which is involved in hepatic glucose metabolism. It controls how much glucose hepatocytes take in and store. High glucose concentrations cause glucokinase activity, which enables the liver to absorb and store more glucose as glycogen when blood glucose levels are high. On the other hand, pancreatic beta cells release the hormone insulin in response to increased blood glucose levels. By enabling the absorption of glucose into various tissues, promoting glucose utilization, and preventing the production of glucose in the liver, insulin works to lower blood glucose levels. Through the metabolism of glucose and the release of insulin, glucokinase and insulin are linked. For the pancreatic beta cells to detect high glucose levels and release insulin, glucokinase activity is essential. When blood glucose levels rise, glucokinase activity results in more ATP being generated, which causes the release of insulin. While insulin encourages the absorption of glucose and storage in peripheral tissues and inhibits glucose synthesis by the liver, glucokinase is involved in the uptake and storage of glucose in the liver. Physiological factors and the type of tissue can affect the glucokinase and hexokinase levels. Hexokinase is present in relatively high concentrations in tissues like muscle that use glucose extensively in order to effectively collect and phosphorylate glucose for energy production. The liver and pancreatic beta cells, on the other hand, have the largest concentrations of glucokinase, where it plays particular roles in glucose sensing and insulin secretion control. These enzymes are regulated differently as well. Hexokinase is control inhibition, in which glucose-6-phosphate, a byproduct of the enzyme, inhibits its activity. This regulation guarantees that glucose metabolism is strictly under control and prevents excessive glucose-6-phosphate buildup. On the other hand, variations in the concentration of glucose regulate glucokinase. Since glucokinase exhibits cooperative kinetics, its enzyme activity rises as glucose concentration does. Because of this regulation, glucokinase can modify its activity in response to changes in liver and pancreatic glucose levels. Let's go back to the beginning of glycolysis. The initial process in the glycolysis pathway is the conversion of glucose into glucose-6-phosphate. Depending on the tissue, either the enzyme hexokinase or glucokinase catalyzes this activity. Let's examine how glycolysis transforms glucose into glucose-6-phosphate. First, Depending on the kind of tissue, glucose enters the cell's cytoplasm where it comes into contact with either hexokinase or glucokinase. By adding a phosphate group from ADP to the glucose molecule, these enzymes aid in the phosphorylation of glucose. Normally, the process is referred to as glucose phosphorylation. The phosphorylation of glucose has a number of significant effects. Hexokinase/glucokinase effectively traps glucose inside the cell by phosphorylating it. Because glucose 6-phosphate can't easily cross the cell membrane, glucose stays inside the cell where it can be used to produce energy. For the regulation of metabolism, the phosphorylation process is also crucial. Hexokinase can phosphorylate glucose even at low glucose concentrations because it has a high affinity for glucose. In contrast, glucokinase functions at greater glucose concentrations and has a reduced affinity for glucose. This distinction enables tissues to control glucose uptake and metabolism according to their individual requirements. An energy investment is made in the glycolytic pathway when ADP is transformed into ADP during the phosphorylation of glucose. It makes sure that glucose is delivered through the glycolytic pathway and not back out of the cell. This first energy investment is later recovered through the glycolysis process. Second, a molecule of ADP is hydrolyzed during the phosphorylation of glucose, releasing one of its phosphate groups. Glucose 6-phosphate, G6P, is created when this phosphate group is transferred to the hydroxyl group at the 6th carbon position of glucose. Depending on the tissue, either the hexokinase or glucokinase enzymes catalyze the process. In general, the phosphorylation of glucose by the enzymes hexokinase or glucokinase is what turns glucose into glucose 6-phosphate during glycolysis. This process controls metabolism, traps glucose inside the cell, and expends energy on the glycolytic route. An essential phase in glycolysis, glucose 6-phosphate goes through a number of enzymatic processes to produce ADP along with other metabolites.